Greetings everyone, and today we are going to discuss about how viruses survive in the host cell. Basically, there are six major ways in viruses survive in the host cell. The first way includes affecting the complement system. We know that in our body there is a complement system which consists of around 20 to 30 uh, complement proteins. These complement proteins are activated whenever there is a virally infected cells within our body. They generally possess a lot of functions such as activating inflammation as well as activating the response of antibodies as well as white blood cells including macrophages and natural killer cells as well as because these are phagocytes or generally we can say of phagocytes as well. Moreover, it also causes to form membrane attack complexes to attack virally infected cells as well as it can also directly form perforations within the cell membrane of the pathogen so that they can damage the pathogens. So accordingly there are many functions for it. They also activate uh, other types of um, cells such as for suppose cell signaling molecules are also activated. The secretion of interferons is also increased uh, by complement proteins by their activation. Now what actually virus does is that it actually uh, inactivates the complement system. Inactivates the complement system and mimics the complement system such that when it mimics the complement system, this mimicking of complement system leads to its inactivation, due to which the complement system is unable to act. After that, second part includes MHC. You might have heard of MHC, major histocompatibility complex, present within our immune cells. The major histocompatibility complex is found in nucleated cells as well as um, uh, MHC1 is present in all nucleated cells except macrophages, whereas macrophages are having MHC2. Now, when it comes to the structure, what is the structure of MHC1 and MHC2? We are going to discuss it in later on in another video. But for now, we are only going to discuss a few things about MHC. That if we are having MHC, some viruses also act on by inactivating the MHC complex. The MHC is the largest gene locus. It is the largest gene locus which contains more than 1000 allele, as a result of which one more thing, the chromosome on which the MHC is located is the sixth chromosome. In human cells, the chromosome on which the MHC is located is the sixth chromosome. Uh, of course, it's an autosome, so sixth chromosome. Now, it is having the largest gene locus, and what virus does is inactivated. As it virus inactivate the MHC, the inactivation of MHC leads the MHC to be inactivated, due to which the cell surface receptors are not found, as a result of which the antigens which are present on the antigen presenting cells are not formed, due to which uh, antigens which are supposed to be recognized by the antigen presenting cells are not recognized, and of course immune response is affected. Example of this virus includes HIV. Apart from that, another example we are having over is the inactivation of interferon response, such that some viruses also act on by inactivating the interferon response in such a manner that these viruses inactivate the release of interferon. What is interferon? It is a cell signaling molecules. These are glycoproteins which resist other path when which when reach to the adjacent cells, cause adjacent cell to inhibit their translation activity. As a result of inhibition in their translation activity, it causes them to resist them against the pathogenic attack of virus, as a result of which this pathogenic activity of virus is inhibited to be spread in other cells. But some viruses also inhibit the interferon spread as well as the synthesis of interferon. Basically, interferons are glycoproteins and these are of low molecular weight. And these are named on the basis of the viral attack, because interferon means something which interferes the activity of virus. These interfere the viral activity by inhibiting the translation activity in adjacent cells, due to which these were known as named as interferon. Now, coming to another point I would like to highlight is the inactivation of B cell. Of course, all of these features are not found in each and every virus. Some virus has a single capability, another virus has another adaptability, but these are the general features. So here, fourth point which I would like to highlight over here is the B cell inactivation. These viruses also cause inactivation of B cell in such a manner that when B cells are inactivated, there is no proliferation, which means multiplication of the B cells, as well as there is no maturation or differentiation of the B cells. As a result of which, there is no formation of B memory cells and B plasma cells, due to which there is no formation of antibodies. As a result of no formation of antibodies, it ultimately causes to inhibit the antibody mediated immune response or the humoral immunity, as a result of which humoral immunity is completely inhibited. One more thing, now this is the a point I wanted to discuss. Now, fifth point here, which I would like to highlight. Here, the fifth point. The fifth point I would like to highlight is that some viruses also uh, alter their genome, which means they sometimes undergo several mutations. Again, there's an example of HIV, that HIV is a virus which always makes mutation. As a result of mutations in its structure, in its viral genome, it many of the drugs are not effective against HIV virus. The same goes with hepatitis C virus, because hepatitis C virus also has a lot of antigenic variations, due to which there is no formation of a particular vaccine or drug against both of these. Moreover, sixth point here includes the that virus have one more thing. That here sixth point, which is how virus survives, is basically sixth point I would like to highlight over here is that viruses basically utilize some sort of strategies such that they wait until they found a favorable condition within the host body, as a result of which they come out whenever there is a requirement. And more than that, some other points I would like to highlight is creation of some enzymes, that some viruses such as bacteriophage has a gene which is utilized to create endonuclease enzyme. That endonuclease enzyme basically degrades the bacterial genome, as a result of which the bacterial cell is under the control of the viral genome, due to which the metabolic machinery are also under the control of viral genome, which leads the bacteria to form protein of its own interest through transcription, translation, and replication. So this is a thing. Now, another thing I wanted to discuss over here is that uh, here we are going to discuss some factors which affect the activity of viruses. Factors affecting viral activity. Now, first factor which I would like to highlight is the temperature. Temperature is one of the major factor, because temperature basically involves a huge impact on virus, such that if temperature is increased, it causes the protein degradation, capsid dissociation, as well as damage to the viral genome. And generally, DNA viruses are more endured, are more stabilized, so DNA viruses have more endurance Then RNA viruses, on the other hand side, RNA viruses are having less endurance because RNA is a less stabilized molecule as compared to DNA, because it is having a hydroxy group at its second carbon, whereas DNA is not having a hydroxyl group. I mean, it's not having oxygen on its second carbon, due to which it is less reactive, while RNA is more reactive. As a result of which, RNA is more likely to cause mutations, as well as is more likely to suffer from high temperature conditions. But if there is high temperature, both RNA as well as DNA viruses uh, have the same degree of damage, which means high amount of temperature makes no difference in damage in them. Moreover, the thing which I would like to highlight is that 60 centigrade, generally 60 centigrade and above temperature can be enough to inactivate viruses, as well as uh, some viruses also have, let's suppose, uh, there are some viruses you know are present within our body, which require certain activities under favorable temperatures. So further temperature, uh, generally 60 and above, can damage them. Moreover, moreover, a thing which I would like to highlight is about the lipid. I mean, the enveloped viruses as well as non-enveloped viruses. Non-enveloped viruses, which are having, they're not having a lipid bilayer, but enveloped viruses which are having a lipid bilayer. First and foremost, enveloped viruses which are having a lipid bilayer are basically having the characteristic of suffering more from high temperature, because envelope, lipid bilayer is more sensitive than non you know than as compared to non-enveloped viruses, because it is having lipid bilayer, having more sensitive to temperature, due to which enveloped viruses are more sensitive to temperature than non-enveloped viruses. Examples of enveloped viruses include influenza and COVID-19, and non-enveloped viruses includes adeno and rhinoviruses, which can also um, secure themselves and endure the heat. I mean, they can also cause infection in the hot temperature as well. One more thing I would like to highlight, which is our second factor, is humidity. Humidity is a factor. First and foremost, let us define what it is. It is the concentration of water vapors present in the air, or the air containing water vapors in invisible form is known as humidity. There are three types of humidity: relative humidity, absolute humidity, and specific humidity. The term which we are going to discuss here is relative humidity. Relative humidity refers to the amount of air, which is amount of water vapors present in air, which is capable to be holded by the air. For suppose if we are having hot air, in hot air, the capability of air to hold the water vapors is higher as compared to the capability of cold air to hold water vapors, as a result of which hot air is having more humidity, more relative humidity, and cold air is having less relative humidity. Likewise, um when it comes to some viruses such as lipid viruses, or we can also call them lipid enveloped viruses, or simply enveloped viruses, are more sensitive. Enveloped viruses such as COVID 19 and influenza are able to, live in only 20 to 30%, able to live in only 20 to 30% of humidity, while non-enveloped viruses are able to live in 70 to 90% humidity. As a result, you can see that as relative humidities increase, there are less amount of viruses who are going to, you know, uh, you know, survive in such a situation, because enveloped viruses are less likely, because they contain lipid bilayer, which is more sensitive to humidity as well as water vapor, due to which enveloped viruses can more suffer from it. Non-enveloped viruses are less suffering from humidity. They can survive in 70 to 90% relative humidity, which includes adeno and rhinoviruses once again. And how is this percentage obtained? It is obtained by the amount of water vapor which water vapor air can hold, divided by amount of total water vapor, into 100, into 100. So as a result of this ratio, we can obtain this percentage. And one more thing, that here humidity is also playing a great role in promoting the growth of viruses in cold air, such that nowadays, as we are having winters, so in winters there's there are more chances of viruses attacking to us, because in winters there is less relative humidity, less capability of air to hold water vapors, as a result of which there is less relative humidity, and more amount of viruses and variety of viruses are able to survive within such an environment and able to infect us, as a result of which more amount of viruses are able to survive within cold temperature. And that is also the reason why we are having many amount of infection cases, many common cold cases, as well as cases of viral infections, and respiratory tract infections within the winter season, because in winter season there is cold air, due to which there is less relative humidity, due to which more viruses are able to survive within the air, and we are then able to we are not able to combat against those viruses, and they cause diseases such as common cold, as well as other sort of diseases. So in this case, this is also the reason. After that, pH, before discussing pH, light, we are now going to discuss about a term, plankton. Plankton is an organism that can survive on the surface of water. Example, phytoplankton such as jellyfish, as well as zooplankton as jellyfish, planktons which animal plankton, zooplankton such as jellyfish, and phytoplankton which includes algae. Examples of these includes ulva. Let me just write that down. Ulva, um, which is also known as sea lettuce, as well as there is anabena. Other example includes chlorella. These are algae. Basically, these are phytoplanktons. After that, uh, um, light energy. I mean, sunlight can be beneficial as well as can be negatively effective for the viruses as well in planktonic environment, in aquatic environment. How? When aquatic environment, as we know that when light energy is, I mean, when light pours over the planktons, it causes to enrich the growth of planktons, as a result of which, as there is more growth of planktons, there are more chances of viruses to have their replication increased within the plankton, because as more furnished and enriched the planktons are, more enriched and furnished would be the growth, replication, or reproduction of the viruses, as a result of which the viruses are then able to increase their growth. And second condition which positively impact them and makes them get an advantage is limiting their attachment with planktons. Basically, plankton's surface proteins are only activated in the presence of sunlight. So when sunlight pours over them, it causes to activate these surface proteins so that the viruses are able to attach with them, as a result of which the planktons are infected and are caused to have their cell surface protein limitized by the attachment of the viruses, due to which the this limiting of attachment is also a key factor behind the positive impact of sunlight. But negative impact of sunlight can lead to, ultraviolet rays. As we previously discussed in our videos, that ultraviolet rays can decay the virus. So negative effect includes the example of UV rays, ultraviolet rays present within the light, which can degrade the virus. Now, coming to our last factor, which uh, I have to discuss over here, is pH. pH is a major factor, such that if we are going supposed to discuss pH, the optimum pH for virus is slightly acidic, 6.5. But if we are going to discuss unfavorable pH in which virus cannot survive, is unfavorable pH includes 5 to 5.5, which is the acidic pH, and 7.2 to 8, which is a basic pH. High basic pH can be dangerous for lipid enveloped viruses because high in basic pH can lead to hydrolysis of the lipid bilayer, as a result of which it can cause to degrade the enveloped viruses. That is also the reason why we utilize sanitizers. Sanitizers as well as detergents are alkaline species. These are having high amount of basic pH, as a result of which when we apply the sanitizers and detergents over our hands, it being at a very high basic pH cause to denature and degrade all of the enveloped viruses present on our hands, as well as also causes the non-enveloped viruses to decrease their rate of proliferation, their rate of reproduction, replication, or multiplication, as a result of which this is the reason why we utilize sanitizer with high basic pH. And yeah, this is all about today's video. Hopefully we'll meet again in another one.